Hello friends, welcome to SRS by tutorial. I am Samanajan. Today we will discuss about epistasis. Epistasis is a type of gene interaction in which one gene pair masks or modifies the expression of another gene pair at different locus. It is an inter-allelic interaction, that is, interaction between alleles of different genes. The gene which masks the expression of another non-allelic gene is called epistatic gene. The gene which was suppressed is called hypostatic gene. This gene interaction modifies the phenotypic ratio of dihybrid cross. Epistasis is classified into 5 types such as dominant epistasis, recessive epistasis, duplicate dominant epistasis, duplicate recessive epistasis, and dominant recessive epistasis. In this video, we will discuss about dominant epistasis and recessive epistasis. Dominant epistasis. If only a single copy of an dominant allele inhibits the expression of either allele of the second gene at a different locus, it is said to be dominant epistasis. It shows a modified phenotypic ratio, that is 12:3:1 instead of 9:3:3:1. Example: inheritance of fruit color in summer squash. There are three colors in summer squash fruit such as white, yellow and green. If a cross was made between a homozygous white squash with homozygous green squash, we obtain plant with white squash in the first filial generation. The intercross between plants of first filial generation produces 12/16 plants with white squash, 3/16 plants with yellow squash and 1/16 plants with green squash. From the above cross, we found that 12/16 or 3/4 plants with white squash and 1/4 plants with pigmented squash. This suggests that a dominant allele at one locus inhibits the production of pigment and resulting in white progeny. If capital W represent the dominant allele that inhibits pigmentation, then genotype for plant with white squash may be capital W or capital W small w, whereas genotype small w allows pigmentation and resulted plants with color squash. Among second filial pigmented plants, there are 3/16 plants with yellow squash and 1/16 plants with green squash. The ratio among pigmented plants is 3:1. This outcome indicates a second locus that determine the type of pigment formation in squash, that is, yellow is dominant over green. This pigmentation is expressed only in homozygous recessive plant having genotype small w. If the genotype has dominant allele of W at first locus, then irrespective of allele Y at the second locus, its phenotype will be white squash. If the genotype has homozygous recessive allele W at first locus, then the dominant allele Y express yellow squash and recessive allele small y express green squash. The presence of dominant allele W masks the color pigmentation in both heterozygous and homozygous conditions. Hence, it is a dominant epistatic gene. In this state, the allele W is epistatic gene, whereas allele Y is the hypostatic gene. Recessive epistasis. If a pair of recessive allele at one locus mask the expression of another gene located at different locus, then it is said to be recessive epistasis. In this condition, the phenotypic ratio of dihybrid cross is modified into 9:3:4 instead of 9:3:3:1. Example: inheritance of coat color in Labrador retrievers. This dog may be black, brown, or yellow. Their different coat color are determined by interaction between genes at two loci. The gene at first locus determines type of pigmentation by skin cell, that is, dominant allele B encodes black pigment and recessive allele B encodes brown pigment. The gene at second locus affect the deposition of pigments in the shaft of hairs. The dominant allele E encodes dark pigment, that is black or brown. The recessive allele E prevent deposition of dark pigment and causing the hairs to be yellow. If a cross was made between a homozygous black Labrador with a homozygous recessive yellow Labrador, we obtain all black progeny in the first filial generation. The intercross between the F1 individual gives F2 progeny in a ratio 9:3:4. The yellow dogs can carry allele for either black or brown pigment, but these alleles are not expressed in their coat color. Therefore, presence of genotype e at the second locus masks the expression of the black or brown allele at the first locus. Hence, recessive allele E is epistatic to allele capital B and small b. Allele capital B and small b are hypostatic to allele small e. As recessive allele E is able to mask the expression of black and brown pigments only at homozygous state, it is a recessive epistatic allele. Another example of recessive epistasis is Bombay phenotype. Bombay phenotype suppress the expression of alleles at the ABO locus. The allele at ABO locus encode antigen embedded on the membrane of RBCs. The allele IA and IB encode different enzymes which add sugar to the end of the carbohydrate chain and designated as A or B. The common substrate on which these enzymes act is H molecule. Most people, a dominant allele at H locus encodes an enzyme that makes H molecule, but in people with Bombay phenotype are homozygous for a recessive mutation that encodes a defective enzyme which is incapable to make H molecule, so that no ABO antigen are expressed on it. Thus, expression of allele at ABO locus depend on the genotype at H locus. Whatever may be the genotype at ABO locus, their expression is totally controlled by the genotype of the H locus. As the H allele masks the expression of allele at ABO locus only at homozygous recessive state, it is a recessive epistatic allele. Thank you for stay connected and watching the video. If you like this video, please share and subscribe to the channel.